Good morning, everyone. It's my pleasure to introduce our distinguished guest today, Professor A.S. Raghavendra, the IOE Chair Professor at the School of Life Sciences, University of Hyderabad. Professor Raghavendra is an internationally acclaimed plant physiologist with over 4 decades of pioneering research work in plant molecular physiology and biochemistry. His research focuses on understanding photosynthesis regulation, stomatal signaling, plant stress physiology, and the interplay between chloroplast and mitochondria. He is renowned for his seminal and research work on nitric oxide signaling and reactive oxygen species, which has transformed our knowledge of how plants respond to environmental stress and also has helped designing new approaches to enhance plant resilience and productivity. He has done his PhD from Sri Venkateswara University, Tirupati, and has worked in Germany as a Humboldt Fellow. Professor Raghavendra is a recipient of J.C. Bose National Fellowship, Government of India, and is a Fellow of INSA, NASI, and NAS. He has also received prestigious recognitions including the INSA Young Scientist Award, Academy of Sciences Young Scientist Award, UGC Career Award, and the INSA Professor T.S. Sadasivan Award. With over 250 influential publications, contributions to landmark books and reviews, and services on editorial boards of leading scientific journals, Professor Raghavendra continues to shape the field of plant science and inspire future generations of researchers. Let us all welcome him with a huge round of applause. Thank you.
Well, thank you very much. First of all, let me thank, I will stand as long as I can, then sit down. First of all, let me thank Professor Rajagopal and Professor Takahashi-san for having arranged this nice meeting, very useful for youngsters and also for us to share some of the thoughts. So today I am going to talk a little bit about actually photosynthetic evolution in eukaryotes. You will not find a very focused kind of approach. I will tell a few points which are interesting, definitely, even for non-plant science people. And whatever I could get from the literature, I try to present here. Oh, maybe you can use that. Well, as all of you know, our Earth has a long history. So what you see here on the left, oops, oh no, it doesn't work. Can you change this to laser pointer? Yes. Mobile, can you change it? So what you see here is millions of years ago, because we are somewhere here, and I am talking of 500 million years ago. What about this? Yeah. Yeah. So life has originated long ago with mostly say like bacteria, and then later in terms of photosynthesis, a group of plants which are called as charophytes. So they are mostly something like between algae and bryophytes, as you can see, chara and related species. So they're all green algae, and from them we have these bryophytes which have originated. Then we have pteridophytes, and later on gymnosperms, and finally angiosperms. As you can see, angiosperms are very, very recent. They have evolved only about 150 years ago. And if you see this Mesozoic and Cenozoic, it has also a lot of how do you call yeah, this I think I will go back later. Now, just to be brief about photosynthetic oxygen evolution. Oh, sorry. I should use that. Yeah, these photosynthetic organisms have evolved with a very interesting kind of background. Oh, okay. Yeah, that may help this also. So we have actually an anaerobic eukaryote, which has engulfed an aerobic bacterium. See, aerobic bacterium can make ATP by using oxygen, whereas this anaerobic eukaryote can give protection to this. So, this has what you call as eaten and kept the aerobic bacterium inside, and then started what you call as evolving. So, you can see as it engulfed, it also developed chlorophyll and ability to make ATP, and so this starts with actually mitochondria. So, when you engulf a mitochondrial cell, it represents the primary symbiosis, and later when a cell has already has this mitochondria, it now engulfs another eukaryote containing chlorophyll, then it becomes what you call as a complete photosynthetic organism. So, this development of mitochondria followed by, this is much more clear. As you see on the top, the host cell will engulf a bacterium which is simple enough, and this bacterium slowly starts developing the kind of inward projections, and which becomes something like very similar to mitochondria what we know of. So, at this stage, this particular microbe with the bacterium inside now engulfs chloroplast. So, if you see with the time, so we have an ancestral prokaryote which is simple, not much differentiation, but then you have this infolding of plasma membrane which helps in developing the nucleus. So, we have a nuclear envelope enclosing nucleus and development of endoplasmic reticulum, and at this stage it engulfs an aerobic heterotrophic prokaryote, and so this is something like a first stage of symbiosis. So, we can say primary symbiosis. Then we have a secondary stage where you get the chloroplast to be engulfed. So, this prokaryote with mitochondria and chloroplast can be now engulfed by another prokaryote. So, this becomes something like a tertiary symbiosis, which I will show you later on. So, we have so stepwise incorporation of organelles, mitochondria like structures, chloroplast like structures, and ultimately getting into a stage where we have the present day what you call as bacterium. So, that is what is represented here. This is all endosymbiosis considered when an aerobic bacterium engulfs an aerobic bacterium, leading to a symbiotic relationship that benefits both. When I say benefit, the aerobic bacterium can make ATP using oxygen, and this is equivalent to mitochondria, and but then this oxygen is not good for the other one. So, it is toxic to the anaerobes. So, this anaerobe provides the ingested nutrients and protection to the engulfed aerobic bacterium. Now, if you see this on a time scale, as I have already mentioned, we have talked of several billion years ago, 3.5 billion years ago, we have a very simple prokaryotes. So, they are all supposed to be represented by the kingdom Monera. They have this very old archibacteria, mostly methane producing, salt loving and heat loving bacteria. Then we have the evolution of typically cyanobacteria. I think you all know that cyanobacteria are the oldest kind of algal systems, very what you call as resistant to harsh conditions in the environment at that time. Of course, then organisms like E. coli, Mycoplasma and so on. And as the time progressed, 2.5 billion years ago, these cells have started engulfing mitochondria and then chloroplast. So, both of them, they become part of eukaryotic cells by this endosymbiotic theory, primary and secondary, and at a later stage it becomes a tertiary symbiosis as well. So, by about 1.5 billion years ago, and even at this stage, we have several of them. Eukaryotes which are small, falling into the category of Protista, and then we have fungi, plantae and animals and so on. And as this evolution progressed, we have some interesting things being developed. So, viruses have developed, and these viruses, since they are capable of infecting several things, some of them are actually what you call as pathogenic, and they don't have a proper DNA, sometimes RNA, never both. So these are actually quite pathogenic, and from here we have even simpler kind of organisms which have only RNA, and later on these also have evolved into something more interesting. You know, proteins are also pathogenic. You must have heard the name of prion and causing bat cow disease. So these are proteins. So we have a whole cell containing nucleus and then organelles. Then we have a cell having either DNA or RNA, and then we have cells which are having only RNA, and then we have organisms which are made of only proteins. So this is a quite interesting kind of evolution going on. And if we go back to the Earth's cycle, as we mentioned, we are talking of Mesozoic era, which started more than 250 million years ago to about 66 million years ago. So this is very old and this is later. So we are talking of minus million years ago. So during this time, we have a Triassic period, Jurassic period, Cretaceous period. I think this Jurassic period, many of you might be familiar with the movie. I think you have seen Jurassic period is very well known for actually the age of reptiles, and these reptiles, what you see in the film is a depiction of dinosaurs, but it doesn't mean only dinosaurs. There are a lot of other organisms as well. But then at the same time, the other side of the story is that there are also a lot of gymnosperms coming up during this time. That's why I think the movie, they tried to depict a lot of these gymnosperms during that time. So when you see the movie, you have to see the science of it. You know, that's more interesting than that. So during this time, we have gymnosperms which are dominant, and dinosaurs which were dominant, and later the flowering plants have started appearing. So during Cretaceous period, instead of gymnosperms, flowering plants became more and more. And at the same time, what you see down here is the Earth itself. As all of you know, at one time the Earth was all one piece of land, and later it started splitting. I think you know about this Gondwana split, quite famous in the history. It breaks down and produces, you know, India on one hand, Africa on the other, South America on the other. And what you see now here is something like this. And then if we go into the recent times, so we are talking of the age at this time. So first of all, it was all animals. So mammalia were dominating. Later, horses and primates have evolved, and then in the so this is called age of mammals. So instead of dinosaurs, we have actually mammals. And during this time, we have also the organisms growing, and from small and like gymnosperms, we have now a lot of land plants evolving, and then there is an evolution of these. Many of these mammals started eating grass and started grazing. So this is quite interesting. Okay, so this is one part of the story. I have been mentioning about the Earth and the life. But when you come to the evolution of photosynthesis actually is quite interesting. We can consider it at the organism level, and we can also consider it at the reaction center level, as all of you know. Typically, what we learn at this time about photosynthesis, I think you will hear more and more of it. We have sometimes photosystem 1, sometimes photosystem 2, both of them in higher plants. But there are organisms where you may have only photosystem 1 or only photosystem 2. So Professor Robert Blankenship is actually one of the authorities on photosynthetic evolution, and he has been trying to describe how the photosynthesis has evolved based on the evidence. And what he said is the earliest organisms were anoxygenic, and that the photosynthetic reaction centers were from a single source. So from this anoxygenic photosynthesis, reaction centers have evolved to become oxygenic, and then they become diversified. So when they become oxygenic, they also developed antenna systems, and then followed by carbon fixation pathways. So reaction centers are the first things to evolve. later diversification of antenna systems, and then the carbon fixation pathways. So, this is actually a kind of evolution. Archaea is a very old one, where you have all the what you call is the type 1. I think this in here this is easier. Okay, this is quite clear. In olden times, we had only purple bacteria, and these purple bacteria were not evolving oxygen, whereas oxygenic phototrophs have developed later, and many of these were using two types of photosystems and two types of reaction centers. This story you all know. Reaction center, photosystem 2 has P680, reaction center photosystem 2 has P700. And please remember, maximum they can absorb is about 700, never more than this. So, we are talking of only red, not far red. But when you look at the earliest organisms, now you can see here this bacteriochlorophyll, which we are talking of in case of purple bacteria, it has only type 2 types of reaction center. It does not evolve oxygen, but it needs very high light. So, something like far red light. I will say this kind of red requiring bacteria are known only in purple bacteria, and a lot of interest on that. And if you look at this connection, as all of you know, photosystem 2 and photosystem 1, they are connected by a cytochrome B6F complex. So, this cytochrome B6F complex has evolved from a different form here. So, if you see here cytochrome B6F complex, it was only BC1 complex. So, this also has evolved, not only reaction centers, but also this connecting cytochrome complex. And at the same time, I should also mention that this type 2 or this type 2 kind of reaction center is based mostly on quinone. I think you must have already heard, we talk of plastoquinone here. So, here also it is a quinone. So, it is a quinone based reaction center or photosystem. Whereas, when you come to photosystem 1, it becomes an iron sulfur type reaction center. So, a lot of details here. And at the other end of the story, you have again a red absorbing kind of reaction center, type 1 type of reaction center, which has iron sulfur type alone. Now, as all of you know, we can have either a non-cyclic electron transport, electrons moving from P680 to P700 and ultimately pyridoxine, whereas you can have only a cyclic type of electron transport with electrons moving from reaction center to pyridoxine and then coming back. So, we have again BC1 complex. So, the evolution has occurred in types of reaction centers, the type of cytochrome B6 or BC1 complex, and then how this electron transport occurs. Yeah, this is what is indicated here, and it is a simple way of representing the earliest reaction center is a very simple one. From here we have the evolution of type 1 RCs, mostly looking like photosystem 1. So, based on the PSA A, PSA B, or we have type 2 reaction centers where you have all this typically D1, D2 and then LM type of things. So, what we have is something like dimers, but heterodimers, whereas in between there are also reaction centers which are quite different from either of these two, and these are also of very high importance in terms of knowledge and in terms of what we call as understanding the evolution of oxygen. In fact, again Professor Blankenship has explained a lot. So, if you want to really know more, you can try to refer this, and in fact, he has written a detailed book on this, but this article is quite good, and as you can see here. Well, let me again mention how the evolution of organelles have happened. I think I have already mentioned, with time the ancestral prokaryote developed membranes, developed nucleus, nuclear membranes, endoplasmic reticulum, then engulfed mitochondria containing or mitochondrial type of the bacterium, and later engulfed something like, it all starts with unicellular algae, and when it engulfs unicellular algae, we have a prokaryote which starts looking like almost like a eukaryote with mitochondria and chloroplast, and then this whole thing sometimes it is engulfed by another one. So we have a first stage of symbiosis, second stage of symbiosis, and then the third stage of symbiosis. Yeah, so this terminology is quite interesting. People try to compare it with the French words. There is a term called as pas de deux, step of two, like what you find in ballet. And what people mean that this evolution of what you call as secondary symbiosis goes very well synchronized. So it's no less than a ballet or maybe even more complicated. The tertiary symbiosis, which you find in many of them, this is even more complex. So it's called Menai-Troyo and a blend of three. So this is a wine with the three types of wines. So they are quite different, but they evolve and work quite well. So this is what is represented here. So primary symbiosis occurred about 1.5 billion years ago. A free living photosynthetic cyanobacterium was engulfed by a heterotrophic eukaryote, which is already an organelle inside, and this now takes in a photosynthetic kind of plastid and now achieves this secondary endosymbiosis. And this one can happen with either green alga or red alga. Both of them are used depending on the situation, but then we talk more about tertiary symbiosis, which is quite interesting. So these events, what we call of a primary symbiosis, so where you have the chloroplast getting engulfed, and then secondary symbiosis, where this is one organism, this is another organism. So as you can see here, both are actually motile organisms, and then they engulf the whole thing. For example, this organism is engulfed by another one, so it becomes tertiary symbiosis. And we have situations of where we have several other instances of tertiary. So let me just read this. Original symbiotic event is believed to have been a primitive flagellate protist and a cyanobacterium. And when it enters, there is a lot of shuffling. Some genes are kept, some genes are lost, and then it results in obligate intracellular chloroplast. Then in secondary symbiosis, we have a heterotrophic straminophile, again motile, and again there is a lot of massive gene transfer and loss of a few components, and then divergence into chromophytes. So chromophytes are all colored organisms. It may be green, or it may be red, or it can be brown, and so on. Then we have the secondary tertiary symbiosis, where a whole dinoflagellate is engulfed and tries to keep up this function of photosynthesis. And people believe that this kind of tertiary symbiosis is still going on. So I will tell you a few examples where we can see this in progress. So what you see here is something like Elysia chlorotica. I think you will be able to understand more. So Elysia chlorotica is a sea slug. It's an animal. It's like a snail, and actually it's quite interesting in several ways. What it does is it eats the brown algal filaments. Vacheria litoria is a typical brown alga, which is what you call a siphonaceous. So it has kind of continuous body. So it tries to eat this and sucks this chloroplast. So what is described during this process is that Elysia chlorotica is a sea slug which sucks or which steals chloroplast from the other host. So we have some details of this coming up. So the chromophytic macroalga Vacheria litoria undergoes symbiosis with E. chlorotica as a result of suctorial feeding. So this sucking itself is quite interesting, and this has been noticed in sea slugs, which are mostly they are all in sea. So then what happens is the nuclei are lost, only chloroplasts are retained, and sometimes these chloroplasts they all get what you call as deposited on the skin, and then this is now quite interesting. As you can see here, this is Elysia chlorotica, which has taken up a lot of chloroplasts, and you can see it keeps this green color, which also means that it is capable of using light and producing certain carbohydrate. And now you can see there are instances in say like our old literature, that there are people who don't need any food, they can simply stand in sun and make a menace, and then they can have their own food making. So very similar and quite fascinating kind of organism. And so this doesn't happen from the birth. So this Elysia chlorotica starts acquiring this chloroplast when it starts eating on Vaucheria. Then we have another example, it's called spotted salamander. As you can see, it is an amphibian, actually quite interesting, because this amphibian can lose parts of its body but still can survive. I think you all know about lizards. Many of the lizards, they can lose their tail but nothing happens, but then this they go to a further level that they can survive. So then here we have what we call as Lotharella globosa, and this is again another interesting kind. Say Paulinella chromatophora is actually eukaryote which engulfs another type of bacterium. I will come to it later. Something like Cercocystis or sometimes Chlorococcum. It engulfs and keeps it during its life cycle. And so we have at least 3 examples which I try to tell in detail. But what you see here in this picture is also diatoms. As you know, diatoms are one of the oldest kind of organisms known to be capable of hosting a lot of smaller algal cells, and they are known to be what we call as symbiotic. And then there is a lot of discussion whether diatoms should be classified into animals or should be classified into plants. So as a plant scientist, we prefer to call them as plants, but then some people claim it as also animals. So now you see here, we try to see a few more details about sea slug, and it will be interesting to see this. ఎందుకు ఇది రావడం లేదా? Oops, sorry. No, it's not working there. రావాలి ఇది. ఇది ప్రెస్ చేస్తే రావాలి కదా. Yeah, this is what happens sometimes. అంటే ఇది పాయింటర్లోకి వెళ్ళిపోయింది. Yeah, now it will come. Yeah, now you can see this is the sea slug we are talking of, Elysia. It's it's trying to feed on this Vaucheria, is a filamentous algae, although it looks green but it's actually brown algae. You can see happily feeds on the algal filaments, and then these algal filaments, they are taken in, and all these chloroplasts, they go and settle finally on the skin. So it is described that it steals chloroplast from the alga and uses it for its own benefit, and for this its mouth parts are specially made for that purpose, to suck the chloroplast from the algal strands. So this goes on for a while, and as you can see, and these chloroplasts are incorporated and they usually find their way onto the skin. So it is believed that these chloroplasts on the surface of the skin, they are significant enough to produce carbohydrates. And this topic is actually a lot of people debate and question whether they can really do this. Some people believe that these chloroplasts are not really sufficient to support the life of this marine slug, Elysia chlorotica. But what is known for sure is that when these plants, they go into something like a hibernation, they can go and survive with minimal kind of eating. So they may be producing some carbohydrate, not to the level of full kind of activity, but capable of allowing survival of the sea slugs. So, these sea slugs have been the topic of study for several people, as you can see. Excellent examples for symbiosis. So, we have an association of a mollusk and then algae, and then trying to what you call as function. So, quite a few of them. Green sea slugs have been objects of curiosity since a long time, as you can see, more than almost like 3 centuries. And then there is a lot of variation, each of this so-called, I have mentioned only one example, but there are other examples. So, all of them, they belong to a group called as Ascoglossa, and each of them has its own kind of choice. So, not every species likes the same species. So, they have their own choice of food like us, and then chloroplast functionality can also vary, but mostly they need something like a siphonaceous type of algae. And then it has also been found that most of these slugs, they prefer to have a brown kind of brown type of algae rather than the green algae. And now when we have this tertiary symbiosis, what you can call them? Is it an animal or is it an alga? Well, I think it is an animal hosting an alga, so it is not an alga hosting an animal. So, but isn't it definitely a good example of tertiary symbiosis. So, this is what I was trying to mention. When the Elysia is born, we can see these eggs, they do not have any chlorophyll, they do not have any chloroplast, they are all plastid free. But as they grow, 21 days also, something like 3 weeks after hatch, they start learning how to feed. So, when they come in contact with Vaucheria, they start what you call as sucking the chloroplast. So, they change their shape, and then after say like a day after feeding, they are at that time they can use this material for their growth, and then this phenomenon is called as kleptoplasty. So, plastids are taken inside and kept closed in the animal. So, that is why they are all called as kleptoplasty, and about 5 days after feeding you can see a nice change in the color. The animal has become green because of this kleptoplasts, chloroplasts which are contained, and the adult one becomes more and more interesting. And as you can see here, we have a lot of plastids. It has grown and quite green, and they are not just green, you can see chlorophyll fluorescing, and if you look at under fluorescence microscope, they can be seen easily. So, they have this chlorophyll fluorescence, and now this is more a lot of more details. There has been a lot of interest in this plant, and it has become possible when scientists have developed a procedure to grow them. As you know, in many of the instances, if you are working with an organism, the moment you are able to grow them in the lab, the research goes on very well. So, that is what you see here. So, you can grow them in aquaria with sea water mimicking solution, but you need to also keep the temperature quite below. So, they have to be maintained about 10 degrees and dim kind of lighting, and you can grow and do a lot of work with this. And this is all examples that chloroplasts are functional. You can put light and then see the evolution. So, this is evolution of oxygen from the Vaucheria, which is a brown alga. You can see a good kind of oxygen evolution system, but Elysia also show this. It responds to light and dark, and then 25%, 100% light and so on. So, the point is that the Elysia chlorotica, which is an animal and which has engulfed and sucked the plastids, it is capable of performing typically the photosynthetic activity. Yeah, the same thing. The cells are not green, then as they start feeding, they what you call as develop green color, and then in this case the alga is slightly different. This is Acetabularia acetabulum. This is also a siphonaceous type of production. Oh, well, actually this figure has a lot of details, and I think we'll try to avoid it. Yeah. Now, a few more examples of the sea organisms which are quite interesting. So, this is another type of sea slug, Sarcoproteus species, and this Sarcoproteus species looks quite similar to an alga which is called as Caulerpa, and you can see this is sea grapes. Okay, Sarcoproteus, another species. This is actually is an alga. Caulerpa is a kind of bryopsidal green alga from coastal regions, and then actually liked by people who are living in the coast, and that's why they are called sea grapes. Yeah, and I think I will consider this as another interesting example. We have what we call as spotted salamander, which is an amphibian, and this is the Latin name, Ambystoma maculatum. It incorporates green algae. What you have seen earlier is brown algae. It incorporates unicellular green algae, for example, Euphila amphilostomaltis, and this alga is very closely related to Chlamydomonas. And let me emphasize that many of these tertiary symbiosis examples, an animal and alga usually are very, very specific in their preferences. Yeah. So, this is salamander, although it can grow actually, and this is as I said, it is known for its ability to survive even if it loses a limb and so on. And now you can see, for example, here this actually shows up a little greenness, although it is not very clear here. But when you look at the eggs, these eggs, they have this unicellular alga quite clear. So, spotted salamanders with single cell algal cells, Euphila amblystomatis, and so it has observed that a lot of interest has been shown to study this, and they found that this chloroplasts, they get incorporated into the cell, and the mitochondria from salamander, they come and try to get around this chloroplast. So, then they have something like a very close association. The chloroplast makes carbohydrates, mitochondria, they also give the energy. So, all of them together, they respire using oxygen and provide carbohydrate and use up together. So, it is something like helping each other. Now, another example, Paulinella chromatophora, it is an amoeba, actually engulfing again unicellular kind of organisms. So, this is a scanning electron microscope, but if you look at under normal microscope, it appears like this, and it is a single cell, and inside you can see chromatophores. So, this has come from the algae, and these algae are actually either Synechococcus or Prochlorococcus. Prochlorococcus is one of the earliest kind of green alga. Synechococcus is earlier one. As you can see, it beautifully stays inside, and both of them they work together. So, unfortunately, it's a bit difficult to grow in lab, unlike the slugs. But then I think one group could grow them, and they have very clearly showed that this chromatophore is quite interesting, and you can analyze the DNA or DNA, and then you can demonstrate that it is of Synechococcus or Prochlorococcus origin. Yeah, this is another example. A freshwater glaucophyte alga is incorporated into Cyanophora paradoxa. As you can see the name, it is quite interesting and quite a paradox to study this animal. And this is again an example of tertiary symbiont. So, this is the Cyanophora paradoxa. It tries to take in a cyanobacterium, tries to incorporate, and can get on with photosynthesis by using both of them together. So, it's we can say it's a model organism where two cyanels or two chloroplasts which are incorporated, and these chloroplasts are called cyanels, and they are quite interesting. And this alga looks like it's very similar to a pathogen, chlamydia, to acquire plastids. So, Cyanophora paradoxa is compared with disease producing pathogen, chlamydia, but it can acquire plastid, and this next figure shows up how this can be done. I think you have already seen how when this mitochondria and chloroplast, they are incorporated through stages: primary symbiosis, secondary symbiosis, tertiary symbiosis, and this tertiary symbiosis going on right now. And now this is an example of, we were talking of chlamydia, Cyanophora paradoxa. This Cyanophora paradoxa is an amoeba type organism. It can take in the what you call as bacteria and chloroplast, and then this chlamydial partner, mostly very similar to this chlamydial pathogens, they are lost during the course of evolution. So, chlamydia are quite infective. So, during the process of evolution of these eukaryotes, they take up this chlamydia type cells, develop archiplastida, and then these are all plants, or something like green kind of thing. But at the same time, chlamydia have evolved in other hosts, as you can see here. They are known to be quite pathogenic. They can infect a variety of organisms, humans, cattle, fishes, mammals, and you can see pigs, birds, even smaller organisms. So, this is that's why they are called as quite paradoxical, to see that chlamydia like bacteria, they can donate to photosynthetic organisms on one hand, and they can continue their life and start and be pathogenic on the other. Yeah, to end up, let me comment about this tertiary endosymbiosis, which has been known in case of volvox. I think volvox you must have heard at several instances. This is a multicellular organism made of actually unicellular organisms. So, it is something like a colony, and then inside this colony we have certain dinoflagellates, and then in these dinoflagellates, some of them are quite interesting. And a good example of this dinoflagellate is a species called as Carenia. Carenia is a dinoflagellate which is famous for producing massive red tides. For example, if you have a chance of going to Florida at a given time, particularly when the temperatures are high, it produces a massive red color. And this massive red color is because of the enrichment of dinoflagellate Carenia, but one has to be very careful. This is quite toxic. So, one should be careful when swimming in these areas. And then why I am mentioning about this Carenia is that this can donate, or it can go into symbiosis and become a part of diatom. So, this again another example which is quite interesting. I think it should be the last one. Thank you, and for your attention.
So, if you have any questions, you can discuss with sir.
Well, evolution is a what you call as fascinating topic. It's very difficult to understand. I don't work on this, just to be frank. I work on the interaction of chloroplast and mitochondria. That's a topic by itself. And I also work on signal transduction in stomatal guard cells.
Any questions?
Either they should have understood completely, or they have not understood at all. Yeah, please. Allowed. Yeah. You have a mic? Yeah.
So in Elixir how we call it a symbiosis? It's
Why do we call?
It's a symbiosis.
Elysia?
Yeah.
Oh, I think you didn't see that earlier slide. Yeah. See, the egg, The egg is colorless, it doesn't have any alga. As it grows, when it comes in contact with Vaucheria, this is a siphonaceous type of alga. They are all continuous tubes. They are not individual cells, not like say like Chlamydomonas. And this can grow only when it comes in touch with this. And as it comes in touch, it starts developing mouth parts and starts what you call as taking up it sucks. And later you can see about 3 weeks after hatch and about 5 days after feeding. So this has taken in chloroplast. So it's basically an animal, but sucks plastids, retains them. And most of these plastids, they go and stay in the surface skin. That's why there is a lot of discussion whether they are all. So if we can have that capacity, if we can humans suck plastids from leaves and retain them. It's not that easy. When you eat, what happens? Everything is digested. So it's a kind of almost like a miracle. It has developed that specialization that chloroplast is not what you call as digested. It is pushed into the skin and retained, and it is retained in terms of function and structure, both. So you have seen that when you look at not here. No. Yeah, there is somewhere I have shown you the chlorophyll fluorescence. So chlorophyll fluoresces, and they can evolve oxygen, and you can see it's almost like the algal cells, some of them. So they are functional plastids are retained.
But for algae?
For algae, well, algae can get a lot of material from the what you call as the animal. Also, algae is also protected from oxygen. Many of these photosynthetic organisms, they also need the proper environment. So some of them, they like this inner kind of atmosphere. Not that it's very ideal, not like they can grow outside also. So it is a kind of both, helping each other.
But the idea I got is they are extracting the plastics.
Extracting and keeping it
For the algae.
intact. Plastid is kept.
Yeah, so for the algae it's harmful, right?
Not exactly. For example, if it is harmful, what about this function? We have we are looking at only chloroplast. The plastid only is evolving oxygen here. If there is already a harm made, how can you expect this activity to go on?
But plastics are
Active. So that means the life is maintained. Plastid life is maintained. If you consider an organelle, whether it is living or non-living, it is living. Yeah. So it must be getting some kind of benefit from the host.
Sir, one question. After the consumption of Eukaryotes, filamentous alga, so does it need any other kind of source, like food source?
Yeah, this is what I said. When you have this green kind of animal, slug, people started arguing, well, it can live, without eating its own food. If you leave it in the sun, it can survive. So a lot of calculations were made, that so many plastids are there on the surface. So these plastids are sufficient. Personally, I believe that amount of food the chloroplasts make is not enough. If you consider the skin surface area and the surface of plastids, you can calculate how much carbohydrate it can make. So that kind of carbohydrate is definitely not enough to make the slugs survive under normal activity. What happens is, as I said, they also have their own ways of what we call as requirement of food. So when the temperature is normal, they move around. They are actually slow moving, so they can try to detect some food around. But if the weather is not very good, it becomes either warm or too cold, they try to settle down on the sea surface and they try to go into something like, I will not say hibernation because hibernation is given for bigger animals. So they try to stay in kind of resting kind of stage. So in kind of resting kind of stage, whatever light they get at the bottom, this chloroplast can back up. Minimum kind of food they can make. So the consensus is that this algal systems in the slug is sufficient to make them survive under kind of inactive period.
Yeah, there's more questions.
So this is,
You can use my.
Yeah.
so this organism is in marine ecosystem, right? So there would
No,
be
I
other-
think your mic is not working.
Yes, what?
Yeah, so I was saying this organism is in marine ecosystem. So there would be other brown algae as well. So why this one is, this sea slug is selective to this brown algae only? Is there a difference in composition of the plastid or both?
Well, that kind of thing is very difficult. For example, nature has evolved in such a way that organisms have likings and dislikings. Suppose I say you can eat every food, you will strongly disagree. When it has evolved, if you are in a different kind of environment, you will eat everything. When you have a chance of saying no, you are saying no. So this specificity, I will try to compare with the similar system in symbiotic nitrogen fixation. In symbiotic nitrogen fixation, the legume has a very specific association with a specific microbe. Actually, microbe not only species, microbe is also species and cultivar stage. So it has such a high degree of specificity. So this liking and disliking has evolved.
Any more questions?
Sir, during the evolution, these plastids, they are retained or every time we need to capture again in individual cycle? These plastids, they are retained in the evolution, in the ratio or
No.
every time?
When this sea slug starts producing egg, they are not retained. Eggs are free of plastids.
Brief life cycle,
Yeah.
it has to.
It has to again start eating Vachyria and acquire. Yeah. Okay, maybe.
Any more questions? Thank you so much, sir, for such an interesting and insightful talk on evolutionary aspects of photosynthesis. We truly appreciate your time and presence and the valuable thoughts you have shared with us. Thank you so much, sir.
Thank you all.